that pairs mentees who are new to ocean acidification work with experienced mentors in the field. The program aims to foster community among ocean acidification professionals, build long-term capacity to measure and address ocean acidification globally, and provide training opportunities to support careers in OA-related fields. Since it began in 2016, Peer2Peer has supported over 250 participants from over 60 countries. If you are interested in participating in Peer2Peer, either as a mentor or a mentee, we will be accepting applications to join the next cohort starting in July 2026, and we encourage you to check out our website to learn more about those upcoming opportunities. Another piece of the Peer2Peer program is we partner with the Ocean Foundation to offer scholarships that facilitate collaborative projects between these mentor and mentee pairs. For any active Peer2Peer participants who are here watching, there is an open scholarship opportunity now with applications due March 31st. So you can reach out to me or see the Peer2Peer website for more information on that. And a couple of words on housekeeping before we get started with today's webinar. During the presentations, all participants are going to be in listen-only mode, and you're welcome to type any questions into the questions box, which can be found at the bottom of the control panel on the right-hand side of your screen. We'll be monitoring these incoming questions, and we'll pose them to our speakers during the question and answer section, which will happen right after each of our speakers. For the discussion, you can also use your raise hand function in the toolbox at the bottom of your screen, and we can call on you to ask your question directly. And with that, I'd like to introduce our first speaker today. Dr. M. Anand will be presenting ARMS, Autonomous Reef Monitoring Structures, an emerging tool to monitor ocean acidification and coral reef health. Dr. M. Anand is an associate professor and the head of the Department of Marine and Coastal Studies at Madurai, Kamaraj University in India. His pioneering research on ocean acidification, coral reef resilience, and marine biotechnology has earned him international recognition and collaborations across Asia Pacific and beyond. Dr. M. Anand is a Peer2Peer alumnus 2023 to 2024 and collaborated as a mentee under the mentorship of Professor Dr. Aileen Tan, Director of the Center for Marine and Coastal Studies at Universiti, Sains, Malaysia. Thank you for joining us today, Dr. Anand, and I will hand it off to you.
Thank you, Dr. Carl, for our nice introduction and coordinating this webinar. Can I share my slides, Carl?
Yes, you can share your slides.
Is my screen visible?
Yes.
Okay. So, very good evening, good afternoon, and good morning to one and all in the different time zones of the participants. I'm Dr. M. Anand. I'm happy to share my experience as a peer-to-peer alumni of Go On, and I thank Go On and the Ocean Foundation and my mentor, Dr. Aileen Hwai, for giving me this opportunity to take up as a alumni of this peer-to-peer. During this program, I acquired some information about the ARMS. ARMS is not new to our ocean acidification research, but ARMS use with reference to the aragonite information and the biological response is what we found it's new to the acidification research. And this technique we got learned from the lab of our mentor, Dr. Aileen in the CEMACS. And I'm going to present my experience, what I acquired some knowledge, and what may be the future research in these aspects of it. The slides are not moving, right?
Yeah, we're not seeing them move.
Wait. I'll do one thing. I'll switch off my video so that buffering can be better. Yeah, is it moving now?
Yes, that's going now.
Yeah. So the journey started with my letter to Dr. Aileen requesting permission to work in her lab, and the the biggest challenge in uh, winning this competitive scholarship is you have to identify the right mentor. Because once you identify the right mentor, only you can write a very good proposal, and you can identify a the right research question. So in that aspects, I wrote to Aileen uh, to learn how to fabricate the arms and how to deploy the arms and how to analyze the results of the arms and all these things within mind. I wrote to Dr. Aileen, and one of the news article which I could come across when I was searching for the mentor is that Aileen has done the first ocean acidification monitoring station. She has uh, started this station in the South China, uh, and this news has provoked me to uh, learn more about these arms, how these arms could be a monitoring tool for ocean acidification research. So that part of the research problem has made me to write to Dr. Aileen, and she readily accepted. Uh, when I written about what type of techniques I wanted to learn from her lab, I elaborated on the what is the background of us and what research gap we wanted to learn from her lab. She readily accepted, and we got the consent from her. So I should thank Dr. Aileen for giving this opportunity. And what I returned to Aileen is, what is the skill set we already have? That is, for the since 2010, we have been doing some microcosm experiments on various calcifying organisms, and uh, recently in the sense by 2021 and 22 onwards, we started monitoring the aragonite saturation state in the Gulf of Mannar and Park Bay regions in the South India. So two different aspects. One area we are very strong in perturbation experiments, and another area we started how the aragonite saturation state can be a signature for ocean acidification monitoring. So between that, what we lack is how the biological organisms responds to it. So that is what we wanted to identify uh, in as a research gap in this particular proposal or the project call, and uh, we could luckily win this call, and we I got paired with Dr. Aileen as a mentee. So what about arms? Arms are, as I told you, it's not new to acidification research. We all know that it's a stackable plates of artificial structures that mimic the natural reef community. So once you deploy these arms near the coral reef region, the organisms will start colonize, will got settle in these arms, so that we will get the information about the organisms distribution, the diversity, and if you could able to collect these information at different sites and different period of time, so we'll get more details about how these cryptic organisms are responding to the changing chemical signatures as the acidification stressors. Is the slide is moving?
Not moving forward right now, but maybe we can give it a second to move.
Yeah.
Yes.
Now, can you see the bridge the knowledge gap? Yeah.
Yeah, we can see that.
Yeah. Yeah. So, what particular research question that we wanted to identify in this particular study was, as I told you, we already done a lot of microcosm experiments in terms of perturbating CO2 in the controlled experiments. And similarly, we already have data on aragonite saturation level. So the biological organisms response, how they react over a period of time, and at what is the succession level when there is changes in the aragonite parameters. This is what we wanted to identify. So for that, we wanted a field-based monitoring for which the arms play an important role. This is where we bridge our knowledge gap. And once this arms has been deployed, we could be able to identify the acidification parameters of aragonite saturation state, alkalinity level, the pH values, salinity, temperature. All these factors will give us an uh, ultimately the value of aragonite saturation state. And the arms factors, from the arms factor, what we could understand is we can linkage how the predictions what we did in the uh, perturbation experiment is that match with the natural condition. So in terms of biomarkers, in terms of DNA methylation, in terms of calcification factors, so all these things we could be able to understand. So this is what we have done, and in our study. This is about the arms materials. Arms material uh, is very simple. Uh, we have a PVC base plates, and these are nothing but the plates are arranged in a regular space with many cross spacers and washers, bolts, nuts, everything. And it's very simple, user friendly. You have in this website the global arms program protocol. One can go through easily, and you can make it a local fabrication also. Similarly, we have ordered from a local agent the PVC plates with all the crossbars, necessary uh, accessories for this arms. We brought the plates, and we assembled the plates in our lab before going for the deployment. This is the place where we deployed the arms. This place is called Pudumadam and Valangamburi. Two places we have deployed these arms. Actually, the study site is a Gulf of Panna region, which is a marine biosphere reserve region, and is known to harbor 3,000 to 600 species of flora and fauna. We have rich coral reefs distributed in this region, and adjacent to this is the Park Bay. Uh, this region also contributes a lot of diversity and supports the diversity of Gulf of Mannar region. And this is the place where we deployed the arms. Once the arms is deployed, we have to give a sufficient time. It depends on the research question what we are going to address. Since we wanted to gain the knowledge, we wanted to gain the experience training of it, and also we had a very short period for this completing this study. We deployed the arms and we have given the time of 46 days to retrieve it back. So once we retrieved, we have photographed the naming and labeling is very very important. And as we extract the organisms, we also taken care to store it for a further sequencing, genome sequencing. Basically, the main structure, I mean, the main result, what we obtained for with this 46 days is we have observed the arms plate in the two different intervals. That is one with the 20th day, and then another time is 46th day. So you can find from the results from when we observe it in the 20th day, it was mostly dominated by bare plate. And you can see the ecological succession very clearly in this, for starting from biofilms is dominating, then cyanobacteria, fungi, then bryozoans, cytozoans, and the slowly colonizing in the 20th day. At the end, 46th day, when we retrieved the arms, we could find the biofilms was there, but equally you can find the succession that cryptic organisms, the grazing organisms are started dominating the plates, and followed by the shrimp larvae, which was seeking the shelter in the arms, and finally the seaweed patches. Seaweed patches, here I wanted to mention, the place where we deployed is called as Seaweed Paradise, the Pudubadam village. So, uh, I mean, at the 46th day, we could find seaweeds, not much of coverage, but we presume that if we leave this arms for another 1 year or 2 year or something like that, we could be able to see the succession very clearly. The ultimately the seaweeds dominate the entire plates. As we took the samples, we have done sequencing. One point I wanted to make a note here is uh, uh, there is a, I mean, the scope for this study is enormous, but within the given duration and within the budget, we wanted to work on it. So we could not be able to go for sequencing of all the organisms what we took from the samples. So selectively, we took three amplicon libraries with molecular markers of koi chain, and we could able to find Cleona, Diadinum, and Aplysia as three organisms we could able to get from the sequencing upon the blast result. Uh, this shows that the organism, I mean, the place is very diverse. We had lot of diverse sequences, but we have preserved it. We wanted to do that sequencing in future and find the diversity. One point here is we have done this uh, observation. Once we have deployed the arms, we started collecting the aragonite parameters level, as well as at the end, during the 20th day and 46th day, we started observing the settlement of the organisms over the plate. What we found is the chemical signatures and the biological response was highly correlating. When we had the alkalinity level as well as the aragonite saturation level was varying, we could find a lot of changes in the plates deposition of these calcifying organisms. Since we have few datas of the sequencing and the aragonate values, I could not able to give you the entire details of it, since we are going for a publication part of it, and doing little part of it is left to go for analysis. So that correlation was existing in this study. When there is changes in the aragonite saturation state, the cryptic fauna settlement is also varied with both the regions, that is the Pudumadam as well as the Valangapuri. The ultimate aim of the study is for the capacity building. So we wanted to this actually the arms usage in India is very limited. A agency called NCSM, which comes under Ministry of Environment and Forest, they started deploying it, and they go for varying modifications of it to studying it. In their website, they have given some details about the usage of arms. Other than this, there is no publications which could be able to support with some data. So what we wanted to present this in the national seminar. Similarly, I've done it in Ocean Society of India conference, which is held in Goa. I could be able to get a lot of response from the scientists, and a lot of informations we shared, and this has given some sensitizing information to the persons who are working in acidification research. And similarly, once our mentor has hosted a program on POGO annual meeting, we showcased our findings in the POGO 26 annual meeting with a general picture about the Gulf of Mannar and Park Bay region, the status of acidification research. In that, we have given our previous studies results on perturbation experiments, aragonate level. We can see in this graph the aragonate levels are given in a map, how the Park Bay and Gulf of Mannar is varying. And the key results, what we achieved out of the study, the scholarship, is we ourselves has got trained with assembling and installing and retrieving and photographing and studying the results of the arms. And apart from this, we have started a regular monitoring in two sites as a PhD thesis work. One of my students started working on it, and the national level workshop, we have trained with 75 participants. The arms results were presented in OSI conference and POGO 26 meeting. The stakeholders meeting was also organized, in which the mentor, Dr. Aileen, has joined with us, and she explained very clearly to the scholars, all the stakeholders who attended the meeting. And the DNA extraction and sequencing has been done, and slowly we are doing for few species as we meet the budget. And another part of this peer-to-peer program is the capacity building, one part, and another is the dissemination of information to the local people. So a lot of local news channel coverage has been done, and also a lot of information has been given in the local newspaper to sensitize this information about the usage of arms, because the use of arms is not only for research. The arms will also, if we could able to deploy the arms in in a large scale near shore, we could able to attract lot of juveniles, brooders, to take these places as a shelter and a feeding ground. So that part has been sensitized to the fishermen, and it was more. I mean, the fishermen were more welcome about this move, and we wanted to take up this further to the local bodies like fishermen organizations, the ministry, to take up this research further. In a long-term study, as I told you, one of my research scholar, actually, as I told you, the study area is a protected area, biosphere reserve region. So formally, we submitted a request to the Ministry of Forest, and they given after our presentation, they given us some recommendations, and with the recommendations, they sanctioned us to allow for only 2 units of arms to be deployed. But again, we wanted to represent to the community to at least 1 unit be increased in an individual site, because for the statistical reason, we want at least 3 arms to be deployed in the same site. So that Mr. Johnson of my lab, he started working on this to identify how this biomarker response and acidification changes can be done in the bivalves. So, what are the challenges and solutions in handling arms? Is the monsoon season, you have to have a clear idea about when the sea is going to be rough and when it will be calm. So, in our area, we had a difficulty in southwest monsoon, so that it made us to delay of deploying our arms. That is the one part, and the budget limitations for DNA sequencing. Lot of scopes is there in this, as you get lot of samples, but DNA sequencing requires lot of budget, so that we have to keep it in mind. And another part is administrative delays, as normally it happens, because the money transfer happens, it has to be university approval has to be done. Due to this, we had a 2 months delay, and luckily I could able to get the 2 months extension from the go on, so that I could complete the results as what is expected for the study. And before I conclude, this is about our department. Our department is in Pudumadam, a coastal village near to the Gulf of Mannar and Park Bay region. But our main campus is in Madurai. We call it a temple city. From Madurai, it is around 140 kilometers to reach our lab. Our lab is a marine field research facility. Actually, it is since we have a very limited facility, we could able to do the wet lab experiments over there. We collect all the samples, we do all the basic processing over there, and we bring it to the main campus, where we have a lot sophisticated instrumentations for analysis. Basically, we have analytical lab and a good facility for seawater pumping, and scuba diving facilities also there. As I mentioned earlier, the Pudumadam village is called a paradise of seaweeds. So we also do the seaweed farming in our place, and we already done that entrepreneurial training. And apart from this, we normally do the International Coastal Cleanup Day every year since 2013. We have taken up an annual movement where students, college students, school students, fishermen community, all they participate, and they do cleanup activity in the Pudumadam village. And this is for the yearly career researchers and students, those who wanted to join as a mentee for this peer-to-peer program. You are very most welcome to our lab. We have a basic facility for microcosm experiments. We have done with organisms of calcifiers. Near our lab, we have bivalves, echinoderms, a lot of diverse organisms are there, so that you can use these organisms to study how these organisms could be respond to acidic conditions. And also, not only the lab based research, if you wanted to do some ecosystem based study, you can very almost welcome, where we have a lot of resources in our labs, near our labs. It's a very rich mangrove ecosystem is there, and coral reefs are there, seagrass, seaweeds. So a lot of resource, or you can see all the ecosystems which are more critical ecosystems of the marine environment is near the vicinity of our lab. And coming to the end, uh, uh, even the mitigation aspects of this acidification also can be studied if you visit our lab, because around our lab we have a lot of seagrass ecosystem and mangroves ecosystem. Recently we published a paper in Springer Nature, in which we have highlighted how this spatial dynamics of aragonite saturation state and the blue carbon stock meadows of the Park Bay is could able to be a mitigating solution for acidification in near the Gulf of Mannar and Park Bay region. So at the acknowledgement, I thank my mentor. I think my mentor is, I'm requested her to be online. If she is online, I thank you, ma'am. Thank you for being my mentor. And I thank Go On and the Ocean Foundation for funding this study. So thank you so much, Carol.
Thank you so much for your presentation, Dr. Anand. That was wonderful. It was really great to hear more about your research, really fascinating results, and also great to hear that it kickstarted some long-term monitoring stations. I hope that some of the peer-to-peer mentees this year or in the
mentees can
future
be
are able to
welcome.
work with you. Yeah.
Sure. We welcome mentees to join here.
Yeah, wonderful. So at this time, we're going to open up the floor for questions. And so you can feel free to put those in the questions box at the bottom right hand of your screen. And we actually have a couple coming in already. So the first question is from Ilaria Dianelio. Thank you for the interesting presentation. I have a question about the ARMS deployment time. Is there a specific reason for choosing 1 month rather than 6, for example? Is it dependent on the site you focus on?
No. Actually, this is not depend on the site or whatever it may be. But to be very frank, the time constraint only, we have deployed the arms and we could able to retrieve within the 46 days. So if the time permits, because we know that peer-to-peer program, it's given for 2 years, right? And with all the administrative formalities and all, and once we start the work, it will be almost 1 year is over. So that is the constraint. But since it was a training opportunity, we did it for 46 months. You can do it for multiple arms. You can deploy, you can retrieve it every 3 months, 6 months, or 6 months after maybe 12 months or 24. Like that, you can partly retrieve the arms so that you will get a very good detail about the succession. And the next question is?
And the next question we have is from Marco Munari from Italy. Great talk, thank you. I was wondering if you considered the effect of light on ARMS colonization, specifically whether you observed differences between the upper panel and the internal plates, and if photophilic versus sciophilic taxa responded differently across the sites with contrasting environmental characteristics.
Yes, it's really an interesting question, Dr. Marco. Actually, this is right. Actually, light aspects is very much important because we have been deploying in a very shallow region, these arms. And as I told you, our place is rich with seaweeds. So we could find in the upper panel a lot of seaweeds are got deposited. And in between that, we could find the tunicates and a lot of ascidians or things like that, which seek the hiding behavior. We could find it within the gaps. So this is, of course, when you see the natural rocks, the same sort of succession we can find it here. The light part is very important. If suppose you deploying the arms in the much more depth, then you will not get the organisms which are being more influenced by the photo, photoic response. So that way, the researcher has to decide what is the depth you wanted to deploy. You can aim for deploying at a different depth, so that you can get a succession of these organisms in very clear.
Excellent. Our next question, well, first you have just thank you for the interesting presentation. Congrats on the achievement. The next question is from Alejandro. What is the required spacing between each plate? And when anchoring them, how deep should they be set?
Yeah, that is very important. Actually, the spacing is very standard in the ARMS protocol. It is there. We can easily find 1 to 1.5 inches is there. And the other part is anchoring is very important. We could find the anchoring depends on the local sea condition. If the sea is much rough, you should have to nail it with the hard surface. And similarly, when you nail it, when retrieving back is very difficult. So depends on the local condition, we have to decide the anchoring facility. Ours, we had a bad experience of first anchoring with the stone, but since it's a rough condition, we lost one arms, and we could able to do it again. So that is what we have. We will get some lessons out of it when you did not understand the local sea condition of it.
Yeah, that's very good advice. Our next question is from Ashanka. Can you explain a little bit more about laboratory mesocosms? What are the pH conditions that you are maintaining, and how you are monitoring changes of organisms there? Thank you.
So actually, that is not a mesocosm. It is actually a microcosm, what we are maintaining, right? Mesocosm is what we need to do it in the wild, in the controlled condition. A part chemical factors or physical factors you can control. But completely what we are doing is a microcosm experiment. Microcosm, we have a perturbation experiment. We have a CO2 cylinder. We purge carbon dioxide into different concentration of at different concentrations to maintain the pH of 8.2, 7.8 and 7.5. It depends on the researcher because previously we have done with echinoderms, we varied it with say 8.2, 7.8 and 7.5 and even 7.2 we have done. So it depends on the researcher, 3 level of pH they maintain or 4 level of pH they maintain for the study.
And I think we have a lot of questions in the chat, but I think we have time for one more. So I'm going to pick one from Amel Balaj. Did you do sequencing for the diversity monitoring or for another purpose? Is there any specific direct relationships between species and acidification?
Absolutely. This is a great question, Amel Belraj, I think, right? Amel. So actually this question, this is what will give a genetic signature of your acidification research. This has to be taken up worldwide. The calcifying organisms will respond in terms of DNA methylation. So if you could able to study the calcifying organisms, the methylation aspects of it by sequencing, you will get a signature of it, so that when you wanted to do a vulnerability study in a particular ocean or something like that, you can collect some organisms, check with the DNA sequencing or signature of the if you have a metadata or a database, we can easily compare of it. So it's a long way to go, but it is the next area of research we should focus on.
Excellent. Thank you for your responses to those, and thanks everyone for your really thoughtful questions. This was a great discussion. If I know that there are a couple people who weren't able to have their questions responded to, so I'd encourage you to reach out to Dr. Anand and keep the conversation going. So with that, thank you so much for joining us today, Dr. Anand. I know that you need to get going, but
thank you. Thank you, Carl. As I told you, I'm on the move. I'll switch off my video and I'll be in the line, right?
Yes, sounds great. Thank you.
Thank you. Thank you. Thank you. Bye-bye.
All right. So at this time, we're going to move on to our next speaker.